Now let us learn about the lactose, which is present in milk and hence called milk sugar. It is composed of two molecules of the monosaccharide, that is beta D plus galactose and beta D plus glucose, where C1 of galactose links to C4 of glucose via a glycosidic linkage, and hence it is known as reducing sugar. So it shows reducing properties. So remember that among the disaccharides, except sucrose, all are reducing sugars. All will be able to reduce Tollens or Fehling reagent. Now let us see the polysaccharides. The polysaccharides are the carbohydrates which yield a large number of monosaccharides on hydrolysis. And we will be learning about the polysaccharide starch, cellulose and glycogen. These are the three types of polysaccharides we will be learning. So, starch, which is a constituent, which is a polysaccharide, is containing two components, two constituents. One is amylose and another is amylopectin. So amylose and amylopectin, you may get a difference between these two components of starch, remember. So the monomer, first of all, let me tell you that the monomer of the starch is alpha D plus glucose, and it contains two components, amylose and amylopectin. Amylose is a linear chain comprising of 200 to 1000 alpha D plus glucose units held together by, you can see, C1 C4 linkages, and it is water soluble and it constitutes 15 to 20% of starch. Whereas on the other hand, if you see the amylopectin, it is a branched chain polymer, unlike the amylose, which is linear chain. It comprises of 20 to 25 glucose units, and it is containing two types of glycosidic linkages, C1 C4 and C1 C6 in the branches. C1 C4 in the linear chains and C1 C6 in the branchings. So this is insoluble in water and constitutes 80 to 85% of starch. So this is the structure of starch, which constitutes the two components, that is amylose and amylopectin. Cellulose is a polysaccharide and it is made up of the beta D plus glucose. The cellulose is a linear polymer of beta D plus glucose, where you can see that C1 of one glucose links to C4 of another via beta links. This is the structure of cellulose. C1 of one glucose is linking to C4 of another via the beta links. So this is all about the polysaccharides, and now we move on. One more polysaccharide I just want to tell you is the glycogen. And glycogen, the monomer is alpha D plus glucose, and it is also known as animal starch because its structure resembles the amylopectin fraction of starch, but it is found in the body of animals, hence it is called animal starch. Now we come to the next biomolecules, and they are amino acids. Amino acids, the term itself indicates amino and acid. That means these are those which contain the amino NH2 in addition to COOH functional group. So those molecules which contain both carboxylic acid and the amino group are called amino acids. Remember, these are the monomers of the proteins, and altogether we have 20 types of amino acids. So on the basis of the amino acids, whether they are synthesized in the body or not, we have two different categories of amino acids. The non-essential amino acids are the ones which are synthesized in the body, example glycine, alanine. Whereas on the other hand, the essential amino acids are the ones which are not synthesized in our body and must be obtained through diet. Remember the example valine and lysine. So this is first classification of amino acid on the basis of whether these amino acids are synthesized in the body or not. Now amino acids, on the basis of their number of NH2 and COOH groups present in them, or on the basis of the chemical nature can be further classified into three different categories. First, acidic, where the number of the COOH groups are more than NH2 groups. Basic, vice versa, number of NH2 group is more than COOH, and neutral, number of NH2 and COOH are same. So amino acids like glycine and valine, which contain one NH2 and one COOH group, are examples of neutral amino acid. Neutral amino acids are those which contain the same number of NH2 basic and COOH acidic functional group. The amino acids like aspartic acid, glutamic acid, which contain one NH2 and two COOH groups, are the acidic amino acid. And those like lysine and histidine, which contain two NH2 groups and one COOH groups, are called basic amino acids. So we have learned about the two classifications of amino acids, and what are these two classifications. These are number one, on the basis of whether they are synthesized in the body or not, and on the basis of their chemical nature. Now let us learn that proteins are the polymers of alpha amino acids. When I say alpha amino acid, what do I mean? To the alpha carbon, that is the carbon adjacent to COOH group, to that carbon NH2 group is attached. So such type of acids are called the alpha amino acids. Now, type of proteins on the basis of the molecular shape. Now, how can we classify the proteins? We can classify them into two different categories depending upon if the polypeptide chains are lying one over the other, parallel to each other, like threads or strands, then such type of proteins are fibrous. Remember, these fibrous proteins are insoluble in water and are held together by strong hydrogen bonds and disulfide linkages. Example of such proteins are keratin, collagen, myosin. Keratin present in nails and hairs, myosin present in muscles, fibrin present in cell. These are the chief structural materials of animal tissues. Now let us see globular proteins. When the polypeptide chain entangle and take a spheroidal or a globular shape, such type of proteins are called globular proteins, where you can see that these are they are held by weak intermolecular forces and hence they are soluble in water. Example are insulin, thyroglobulin, albumin, hemoglobin, fibrinogen, and all these are examples of globular protein. In clotting of blood, fibrinogen gets converted to fibrous protein, fibrin. Remember. Next is, let us learn the structure of proteins. Proteins have the four different structures: primary structure, secondary structure, tertiary structure, and quaternary structure. Primary structure of protein tells us about the manner or the sequence in which the amino acids are arranged in the polypeptide chain. In what specific sequence are the amino acids arranged in the polypeptide chain? This idea is given by the primary structure. Any change in the sequence of even one amino acid creates altogether a different protein with different biological function or different properties. For example, the hemoglobin protein contains 574 different types of amino acid. If there is change in the sequence of even a single amino acid, then the biological properties of this hemoglobin protein are destroyed and the RBCs become sickle shaped, causing a disease called sickle cell anemia. Now we come to the secondary structures of protein. What are secondary structures? It refers to the shape in which long polypeptide chain exists. The long polypeptide chain may acquire or may take a right-handed helical shape, or they may get stacked one over the other, forming a beta pleated structure. So the two secondary structures of protein, remember, are the alpha helix and the beta pleated structure. The alpha helix results due to the regular coiling of polypeptide chain and contains intramolecular hydrogen bond. The basic difference between the two structures are that the alpha helix contains intramolecular hydrogen bond, whereas the beta pleated contains intermolecular hydrogen bond. Keratin in hair, nails, wool and myosin in muscles have the alpha helix structure. Now let us study about the tertiary structure of proteins. The tertiary structure of protein arises as a result of overall folding of the polypeptide chains, and the tertiary structure of protein is of two types, fibrous and globular. So tertiary structure arises as a result of folding of the secondary structure, and the forces which stabilize the secondary and the tertiary structures of proteins are the hydrogen bonds, disulfide linkages, van der Waals forces and electrostatic forces of attraction. Now, after tertiary, let us learn what are the quaternary structures of proteins. Some of the proteins are composed of two or more polypeptide chains called subunits. The spatial arrangement of these subunits means the three-dimensional arrangement of these subunits with respect to each other is known as the quaternary structure. So, the primary structure tells us about the sequence of amino acid. By the further folding of the primary structure, the secondary structure arises, which is of two types, alpha helical and beta pleated. By the further folding of the secondary structure, the tertiary structure arises, which is of two types, fibrous and globular. And when the subunits of protein, they acquire a three-dimensional arrangement, they give rise to the quaternary structure. That means from tertiary structure, the quaternary structure arises. Now, a very, very important topic, and that is the denaturation of proteins. Now, denaturation of protein can come as a short note or maybe as a question in your board examination, short note question in your board examination carrying 1 mark. So, what is denaturation? The protein in its original state is called the native state of protein. When that native state is subjected to a change in the temperature or in the pH, then the hydrogen bonds, they get disturbed. The primary structure remains the same, remember, but only the secondary and the tertiary structures are destroyed, but the primary remains intact and the protein loses its biological activity. In this process, the globules unfold and the helix get uncoiled and the protein loses its biological activity. This is what is meant by denaturation of protein. We have two examples of denaturation, that is coagulation of egg white on boiling and curdling of milk as a result of fermentation. So, denaturation of protein is a very, very important topic. Now we come to enzymes. What are enzymes? Enzymes are the biological catalyst which catalyze the biochemical reactions taking place in the body, and enzymes, remember, are globular type of proteins. So here, for example, the enzyme working on maltose will be maltase. For example, the enzyme working on the sucrose and breaking down into glucose and fructose will be invertase. For example, the enzyme working on the lactose will be lactase, and depending upon on which substrate do they work, they are characteristic in their action, and we have different names for different enzymes. Enzymes, you have to remember, are globular type of protein. Let us see a few examples of enzymes and their roles. Oxidoreductase enzyme are the ones which catalyze the oxidation of one substrate simultaneously reduction of another substrate. Phenylketonuria is a disease which is caused by the deficiency of enzyme phenylalanine hydroxylase. When this enzyme phenylalanine hydroxylase becomes deficient, it causes a disease called phenylketonuria, PKU. The enzyme tyrosinase, its deficiency causes the disease called albinism. And streptokinase is the enzyme which dissolves the blood clot formed in coronary artery, leading to heart attacks. So this is all about enzyme. Learn a few examples of enzymes, and we will already be learning about enzymes in detail in the unit surface chemistry. Now let us learn about the nucleic acids. Nucleic acid is another category, another third category of the biomolecules, and they are the polymers of the nucleotides. Nucleotides are their monomeric units. So we will discuss about two different types of nucleic acid, DNA and RNA. Now let us see what are the constituents of nucleic acid. There are three. This can come as a 1-mark question. On hydrolysis, nucleic acid result in the formation of three components: pentose sugar, phosphoric acid and nitrogenous base. You have to remember that nitrogenous bases are of two categories: pyrimidines and purines. Pyrimidines contain the bases cytosine, thymine and uracil, and purines contain the bases adenine and guanine. Remember that in RNA the thymine is replaced by uracil. Nucleoside. If you see what is a nucleoside, then a nucleoside is a component of nucleic acid which is formed by the attachment of the sugar to the nitrogenous base. So a unit formed by attachment of base to 1' position of sugar is called nucleoside. But when to a nucleoside phosphoric acid links at 5' position of the sugar, then it results in the formation of the unit called nucleotide. That means nucleotide minus phosphate is nucleoside, and the monomers of the nucleic acid are nucleotides, which are formed by three components, that is the pentose sugar, phosphoric acid and nitrogenous base. Excluding phosphoric acid, that means the combination of pentose sugar and nitrogenous base is called the nucleoside, to which if phosphoric acid attaches at the 5' position of the sugar, then it results in the formation of nucleotide. Now we will learn about the nucleotides. They are joined by the phosphodiester linkages. This comes as a 1-mark question. Phosphodiester linkages are the linkages between the 5' and 3' carbon atoms. Remember, the linkages in case of carbohydrates are glycosidic linkages. In case of proteins are peptide linkages. In case of nucleotides are phosphodiester linkages between 5' and 3' carbon atom of the pentose sugar. So you can see the phosphodiester linkages resulting in the formation of a dinucleotide. Now let us learn about the nucleic acid DNA. DNA, the full form is deoxyribonucleic acid, and the components of DNA are pentose sugar deoxyribose, nitrogenous base adenine, guanine, thymine and cytosine, and a phosphate group present or attached to C5 of the sugar unit. The DNA has a double helical structure, and the two strands of DNA are said to be complementary to each other. This can come as a 1-mark question. Why the two strands of DNA are said to be complementary and not identical? Remember, if on one strand there is adenine, on another strand there will be thymine. On one strand if there is cytosine, on another strand there will be guanine. So remember that there is a base pairing principle which says that adenine links to thymine via a double hydrogen bond and cytosine to guanine via a triple hydrogen bond. So suppose there is a base sequence stating that on one strand of DNA ATG, then on the another strand the base sequence will be, in front of A there will be T thymine, in front of T there will be A adenine, in front of C there will be G guanine. So these are the complementary bases, that is A and T adenine and thymine, and G and C guanine and cytosine. So that is why we say that the two strands of DNA are not identical but complementary. This can be explained on the basis of the base pairing principle. DNA, remember, is present in the nucleus and is responsible as a genetic material responsible for transmission of the hereditary characters from parents to offsprings. So DNA can replicate. This is another important feature of DNA. Now we come to the next nucleic acid, and that is the ribonucleic acid, RNA. RNA, the components are ribose sugar. It contains the bases uracil, cytosine, and the adenine and guanine. That means in DNA, the thymine is replaced by the uracil in RNA. So a phosphate group is again there at the C5 of the sugar unit, and the repeating units are called ribonucleotides. It contains a single helical structure, and the major function of the RNA is the synthesis of protein. The site of protein synthesis is ribosomes, and the two steps of protein synthesis are transcription and translation. Remember that RNA is present in the cytoplasm of the cell, and it is a single stranded structure. It cannot replicate, rather it is formed from DNA. It is of three types: messenger RNA, transfer RNA, and ribosomal RNA. Messenger RNA called mRNA, transfer called tRNA, and ribosomal called rRNA. Now, functions of nucleic acid. As just now we discussed that DNA is responsible for the transmission of hereditary characters from parents to offsprings, and also the process by which a DNA molecule produces two identical molecules of itself is called replication. So replication, it is responsible for replication or hereditary transfer. So it's a genetic material, remember. Protein synthesis, it is the function of the RNA, and it is done in two steps: transcription and translation. So you need not go into the details of these two steps, transcription and translation. And now we move on to the next category of biomolecules, which are vitamins. Vitamins are, if you break this term vitamins, the literal meaning of vitamins is vital amines. These are those organic compounds which are required in diet in small amounts and trace amounts, and are responsible for performing special biological functions for the normal growth and maintenance of the organism. So these are the vital amines. They are required in very trace amounts, and their deficiency causes certain diseases in the organisms that we are going to discuss in detail, which can come as a 1-mark question in your board examination. So depending upon their solubility or in fat or water, we can classify them into two categories: fat soluble vitamins and water soluble vitamins. Remember, A, D, E, K are fat soluble vitamins. They are insoluble in water, and they are stored in liver and the adipose tissues. Whereas the vitamins B and C, except B12, are the water soluble vitamins. A question comes for 1 mark that why the vitamin B and C should be regularly supplied in the diet. It is so because they are water soluble and are readily excreted along with the urine and cannot be stored in our body. So because these vitamins are flushed off with the urine, hence they should be regularly supplied into our diet. Now this is a table which will tell you about the sources of all the vitamins and the deficiency diseases caused by these vitamins. So let us study some important vitamins, their sources and deficiency diseases, which I am sure you have learnt about few of them in your lower classes as well. So vitamin A, which is the rich source, is fish liver or cod liver oil, carrots, butter, milk, etc. The deficiency disease it causes is the night blindness or xerophthalmia, which refers to the hardening of the eye cornea. Next is vitamin B1, chemical name is thymine. Vitamin A, chemical name is retinol, retinol. B1 name is thymine. Present in yeast, or the sources are yeast, milk, green leafy vegetables and cereals. Deficiency causes beriberi. Symptoms of beriberi are loss of appetite and retarded growth. Vitamin B2, chemical name riboflavin. Present rich sources are milk, egg white, liver, kidney, etc. Deficiency causes chelosis, which results in fissuring or cracks at the corners of the mouth and lips, digestive disorder, and burning sensation in the skin. Vitamin B6 is pyridoxine, which is the rich sources are yeast, milk, egg yolk, cereals, and grams. The disease caused by this is convulsions. Vitamin B12, the chemical name is cyanocobalamin. Cyanocobalamin, and the sources are meat. That means all the non-veg items. That is meat, fish, egg, and curd. And the deficiency disease caused by this is pernicious anemia, where the RBC becomes deficient in hemoglobin. Vitamin C chemical name ascorbic acid. C for citrus fruits. So all the citrus fruits are rich in vitamins. The richest source of vitamin C is avla and green leafy vegetables. And the deficiency diseases is scurvy. The symptoms are bleeding gums. Vitamin D. D is the sunlight is the rich source of vitamin D. Apart from that, fish and egg yolk. The chemical name is ergocalciferol. Ergocalciferol. Then the deficiency disease is rickets, which results in bone deformities in children, and osteomalacia, where the bones become soft and fragile, and joint pains in adults, leading to arthritis. Vitamin E, tocopherol. Vitamin E chemical name tocopherol. Vegetable oils are the rich sources like wheat germ oil, sunflower oil, etc., sesame oil. The deficiency disease, increased fragility of RBCs and muscular weakness. Vitamin K, phylloquinone, green leafy vegetables, it increases the blood clotting time, delays the blood clotting, hence it is also called anti-hemorrhage vitamin. So children, this is all about all the biomolecules we have studied in this chapter. Prepare this chapter thoroughly from your NCERTs and the video, and try to solve the NCERT back exercises. Thanks for watching. Have a nice day.